Health officials and virologists are intensifying their focus on a rare but high-consequence respiratory pathogen that remains a significant concern for residents in various regions of the United States. Recent reports highlighting the dangers of domestic dust exposure have brought renewed attention to the hantavirus, a family of viruses primarily spread by rodents. While the overall number of cases reported annually remains low, the severity of the illness, known as Hantavirus Pulmonary Syndrome or HPS, necessitates a high level of public awareness. The primary risk factor identified by clinical studies and epidemiological surveillance is the inhalation of aerosolized viral particles, often occurring during the cleaning of enclosed spaces like attics, basements, and sheds. This lethal dust phenomenon occurs when dried rodent excreta, including urine, droppings, and saliva, are disturbed and become airborne. Once inhaled, the virus begins its incubation period, which can range from 1 to 8 weeks, though most cases manifest symptoms within 2 to 3 weeks of exposure. The gravity of the situation is underscored by the high mortality rate associated with the virus, which the Centers for Disease Control and Prevention estimates at approximately 38%. This makes HPS one of the most lethal respiratory infections currently monitored by public health agencies. Geographically, while cases have been reported in various parts of the country, the western United States has historically seen the highest concentration of infections, particularly in the Four Corners region, where the virus was first identified in 1993. However, the reservoir hosts, such as the deer mouse, are found throughout North America, meaning the potential for exposure exists far beyond documented hotspots. Understanding the intersection of environmental conditions and human behavior is critical for framing the current threat landscape. As homeowners engage in seasonal cleaning or renovation projects, the risk of disturbing long-settled dust in poorly ventilated areas increases. Public health reporting emphasizes that even a single significant exposure event can lead to infection, challenging the misconception that prolonged contact with rodents is necessary for transmission. This report will examine the specific biological mechanisms that make this virus so dangerous, the clinical challenges faced by healthcare providers, and the coordinated efforts by surveillance agencies to track and mitigate the spread of this zoonotic threat. At the core of the clinical challenge is the rapid progression of Hantavirus Pulmonary Syndrome, from mild flu-like symptoms to life-threatening respiratory failure. In the early stages, patients typically report fever, severe muscle aches, particularly in the large muscle groups of the thighs, hips, and back, as well as fatigue and chills. Because these symptoms mirror common seasonal illnesses, early diagnosis remains a significant hurdle for hospitals and clinics. Diagnostic labs rely on specialized testing, including enzyme-linked immunosorbent assays to detect hantavirus-specific antibodies and polymerase chain reaction, or PCR, to identify the viral genetic material itself. However, these tests are often performed at state public health laboratories, or the CDC, which can lead to a delay between the onset of symptoms and definitive confirmation. Once the virus enters the lungs, it targets the endothelial cells that line the blood vessels. This leads to a systemic inflammatory response, where the capillaries begin to leak fluid into the alveolar spaces of the lungs. This stage, known as the cardiopulmonary phase, is characterized by a sudden onset of shortness of breath and a cough that progresses rapidly to acute respiratory distress. Unlike many other viral pneumonias, HPS does not typically cause the long-term cellular destruction seen in other infections. Instead, the primary issue is the volume of fluid preventing oxygen exchange. Medical facilities emphasize that there is currently no specific cure, vaccine, or antiviral treatment for hantavirus infection. Patient management is limited to supportive care, which often involves intubation and mechanical ventilation in an intensive care unit. In the most severe cases, extracorporeal membrane oxygenation, or ECMO, is used to oxygenate the blood outside the body, while the lungs attempt to recover. Public health agencies like the CDC and the Occupational Safety and Health Administration monitor these clinical outcomes to refine their guidance for at-risk populations. Surveillance data indicates that while most cases are sporadic, clusters can occur when environmental conditions favor an explosion in the rodent population, such as periods of increased rainfall following a drought, which leads to a surplus of food sources for deer mice. This ecological link makes the work of field biologists and wildlife agencies essential to the overall public health response, as they provide the data necessary to issue timely warnings to specific communities. Prevention remains the primary tool in the global effort to reduce hantavirus mortality, with public health guidance focusing heavily on the safe management of environments where rodents may nest. Official statements from health departments stress that individuals should never use sweeping or vacuuming methods to clean areas where rodent activity is suspected, as these actions are the most likely to aerosolize the virus. Instead, the standard recommendation is to thoroughly wet the affected area with a disinfectant solution, such as a mixture of 1 part bleach to 9 parts water. By saturating the dust and droppings, the viral particles are neutralized and kept from entering the air. Experts also advise the use of protective gear, including rubber gloves, and in high-risk scenarios, N95 or higher-rated respirators to ensure that no particles are inhaled during the cleaning process. Beyond immediate cleaning protocols, long-term prevention relies on rodent-proofing structures. This involves sealing gaps larger than a quarter inch, removing food sources, and keeping vegetation clear of building foundations. On a broader scale, surveillance programs continue to track the prevalence of the virus in wild rodent populations to identify emerging risks. While the sin nombre virus is the primary concern in the United States, global health organizations monitor related hantaviruses in Europe and Asia, such as the Pumala and Hantan viruses, which cause hemorrhagic fever with Raynaud syndrome. Although the clinical presentation differs, the underlying transmission mechanism remains the same: human contact with infected rodent waste. There remain significant limits to our knowledge, particularly regarding why certain individuals develop more severe immune responses than others, and the exact duration the virus remains viable in different environmental conditions. Researchers are currently investigating the genetic factors of the virus and its hosts to better predict future outbreaks. Until a vaccine or targeted therapy is developed, the burden of protection lies in education and meticulous environmental hygiene. Public health officials urge anyone who develops a sudden fever and shortness of breath after cleaning a potentially infested area to seek medical attention immediately and to inform the provider of the potential exposure. This transparency in clinical reporting is vital for ensuring that supportive care begins as early as possible. For more updates on public health developments and emerging viral threats, consider subscribing to this channel. The core of the clinical and scientific challenge surrounding hantavirus lies in the rapid, often catastrophic progression from initial inhalation to full-scale respiratory failure. While the phrase "one breath" may seem like hyperbole, virologists and agencies like the Centers for Disease Control and Prevention confirm that the virus is remarkably efficient at infecting the human host through the aerosol route. When dust in an attic or crawl space is disturbed, microscopic particles of dried rodent urine, saliva, and feces are suspended in the air. These particles, which may carry the sin nombre virus, the primary strain found in the United States, are small enough to bypass the upper respiratory defenses and settle deep within the alveolar sacs of the lungs. Once the virus enters the body, it initiates an incubation period that is notably silent, often lasting between 1 and 5 weeks. During this window, the virus replicates within the endothelial cells, which form the lining of the blood vessels. Unlike many other viral pathogens that cause direct cellular destruction, hantavirus triggers a massive localized immune response. This leads to a dramatic increase in capillary permeability, effectively causing the blood vessels to leak plasma into the lungs. This process, known as pulmonary edema, is what leads to the extreme risk of death mentioned in recent health alerts, as the patient effectively drowns in their own bodily fluids. In a clinical setting, this transition from a prodromal phase of fever and muscle aches to the acute cardiopulmonary phase can occur in a matter of hours, leaving medical teams with a very narrow window for intervention. Hospital systems and diagnostic laboratories face significant logistical hurdles when dealing with suspected hantavirus cases. Because the early symptoms are indistinguishable from common influenza or other seasonal respiratory viruses, hospitals often do not suspect hantavirus until the patient's oxygen levels begin to plummet. At this stage, the diagnostic process becomes a race against time. Specialized laboratories utilize enzyme-linked immunosorbent assays, or ELISA, to identify the presence of IgM antibodies, which indicate a recent and active infection. Additionally, molecular testing through polymerase chain reaction, or PCR, is used to detect the viral RNA itself in blood or tissue samples. However, because these tests are technically demanding and require high containment biosafety protocols, many local hospitals must ship samples to state public health laboratories or the CDC's Viral Special Pathogens branch. This logistical chain can introduce delays of 24 to 48 hours, during which time the patient's condition may deteriorate. To counter this, official medical guidance emphasizes that doctors should prioritize supportive care based on clinical suspicion and history of exposure rather than waiting for laboratory confirmation. There is currently no FDA-approved antiviral drug proven to be effective against Hantavirus Pulmonary Syndrome, ribavirin, while used in some cases globally for other hantaviruses, has not shown a significant clinical benefit for the respiratory form seen in North America. The scale of the extreme risk described by officials is also reflected in the complexity of surveillance and environmental monitoring. Agencies such as the National Institute of Allergy and Infectious Diseases monitor the ecological factors that lead to increased human risk. They track the population density of the deer mouse, or Paromyscus maniculatus, which serves as the primary reservoir for the virus. When environmental conditions such as high rainfall lead to an abundance of seeds and insects, rodent populations can surge, increasing the likelihood of home infestations and the subsequent accumulation of infectious dust. Verified data from the CDC indicates that the virus can remain stable in the environment for several days depending on temperature and humidity, though it is highly susceptible to sunlight and standard household disinfectants. This environmental fragility contrasts with the lethality of the virus once it enters the human system, creating a situation where the pathogen is rare but disproportionately dangerous. Public health reporting continues to emphasize that while the total number of cases documented across the United States remains relatively low, often fewer than 50 cases per year, the mortality rate of nearly 40% elevates hantavirus to a top-tier priority for infectious disease surveillance. The focus remains on bridging the gap between field ecology, where the virus originates, and the intensive care units, where the final battle for the patient's life takes place. The primary defense against hantavirus mortality is a shift in how individuals interact with potentially contaminated environments, moving away from traditional cleaning methods towards specialized, biohazard-aware protocols. Public health officials emphasize that the most critical error a homeowner can make is the use of dry cleaning methods, such as sweeping or vacuuming, in areas where rodent activity is evident. These actions do not remove the threat. Instead, they serve as the catalyst for aerosolization, lifting the dried, viral-laden organic matter into the breathing zone of the individual. To mitigate this lethal dust risk, the established clinical standard for remediation involves a process of heavy saturation. Before any cleaning begins, the area must be ventilated for at least 30 minutes to allow any existing airborne particles to settle or be dispersed by fresh air. Once ventilated, all droppings, nests, and contaminated materials must be thoroughly soaked with a disinfectant solution, typically a mixture of 1 part bleach to 9 parts water. This contact time is essential, as the chlorine solution breaks down the lipid envelope of the virus, rendering it inactive before it is physically moved. Only after a 10-minute soak should the materials be wiped up using paper towels or rags, which must then be double-bagged and disposed of in a secure outdoor trash receptacle. This meticulous approach is the only verified way to prevent the accidental inhalation of the pathogen during domestic maintenance. Beyond immediate cleaning, the long-term strategy for public health focuses on structural exclusion and the management of the domestic perimeter. Experts in zoonotic disease prevention advise a seal-up, trap-up, and clean-up methodology. This begins with an exhaustive inspection of the home's exterior to identify any gaps larger than a 1/4 inch, which is the minimum space required for a deer mouse to gain entry. Using materials that rodents cannot easily chew through, such as steel wool or metal flashing, is a standard recommendation for sealing these breach points. Simultaneously, the removal of attractants, specifically unsecured pet food, birdseed, and improperly stored grains, is vital to reducing the local rodent population density. Outside the home, clearing heavy brush and wood piles away from the foundation removes the nesting sites that serve as the reservoir's base of operations. These practical steps represent a shift from reactive medicine to proactive environmental management, recognizing that in the absence of a vaccine or specific antiviral treatment, the most effective way to save lives is to prevent the spillover of the virus from the wild rodent population into the human domestic sphere on a global scale. Hantavirus surveillance remains a complex and resource-intensive endeavor coordinated by agencies such as the Centers for Disease Control and Prevention and the World Health Organization. These organizations maintain a network of sentinel sites where rodent populations are regularly trapped and tested for viral prevalence. This data allows public health officials to issue regional warnings when the percentage of infected rodents reaches a critical threshold, often following specific climatic events like heavy rainfall that lead to a surplus of food and a subsequent population explosion. However, significant limits to our knowledge remain. Scientists are still investigating why some regions see higher rates of the pulmonary syndrome while others see the renal syndrome, and the exact biological triggers that lead to the cytokine storm in the human lungs are not fully understood. There is also the ongoing challenge of diagnostic speed. Until rapid point-of-care testing becomes widely available in rural clinics, the burden of early detection will continue to fall on the patient's ability to report potential exposure to their healthcare provider. The ethical imperative for public health reporting in this context is to balance the gravity of the 38% mortality rate with a calm, evidence-based call to action. While the risk of a single breath of contaminated dust is medically verified, it is also a preventable tragedy. The goal of providing this information is not to incite panic but to equip residents with the specific technical knowledge required to navigate high-risk environments safely. As long as hantaviruses exist in their natural reservoirs, they will continue to pose a threat to those who disturb long-settled dust in forgotten corners of their homes. Honest communication about the limits of current medicine, combined with rigorous adherence to proven prevention protocols, remains our most reliable tool for reducing the impact of this lethal pathogen. For ongoing updates on emerging health risks and detailed reporting on global disease surveillance, consider subscribing to this channel.